This is the second session that describes the control and regulation of respiration. The learning objectives are to explain the regulation of respiration, to describe the chemical regulation of respiration, and to describe the clinical application of today's session. Starting with the regulation of respiration. We all know that the respiratory center activity is regulated by impulses coming from large number of sensors along afferent nerves according to the body needs. There are two main classes of sensory systems conveying information about the function of the respiratory system: the chemoreceptors and the other non-chemical receptors. Accordingly, there are two modes of regulation of respiration: chemical regulation and non-chemical regulation. Chemical regulation, which involves the effect of variation of the blood chemistry, that's to say, the partial pressure of oxygen, partial pressure of carbon dioxide, and the hydrogen ion concentration on the ventilation. It's mediated through the chemoreceptor, and it's the basic mechanism for regulation of rhythmic breathing. While the non-chemical regulation is superimposed on the basic chemical regulation of respiration, it involves other afferent that provide non-chemical regulation that affect the breathing in particular situation. In this session, we will discuss the chemical regulation of respiration. The chemical regulation of respiration is the basic mechanism for regulation of breathing. The chemical regulatory mechanism adjusts ventilation in such a way that the partial pressure of carbon dioxide is normally held constant, the effects of excess hydrogen ion concentration in the blood are reduced, and the partial pressure of oxygen is raised when it falls to potentially dangerous level. The effect of changes in the arterial blood gases of the partial pressure of oxygen, partial pressure of carbon dioxide, and hydrogen ion concentration on ventilation is mediated through the respiratory chemoreceptors. A chemoreceptor is a receptor which monitors any change in the chemical composition of the blood or the surrounding fluids. They respond by reflex changes in the ventilation that return the arterial blood gases towards normal levels. There are two types of respiratory chemoreceptors: central chemoreceptors and peripheral chemoreceptors. An increase in the partial pressure of carbon dioxide or hydrogen ion concentration, or a drop in the partial pressure of oxygen in the arterial blood, increases the activity of the respiratory center and thus increases the pulmonary ventilation. Changes in the opposite direction have an inhibitory effect on the ventilation, and in this way, the respiratory rate is adjusted according to the metabolic rate. Starting with the central chemoreceptors. The central chemoreceptors are located bilaterally just beneath the ventral surface of the medulla. These chemoreceptor neurons are anatomically separate from, but synaptically communicate with, the neurons of the respiratory control center in the medulla. Because of their location inside the brain, they are protected by the blood-brain barrier, which separates the blood from the CSF. They respond to change in the pH of the brain extracellular fluid and the cerebrospinal fluid. Now, what is the response of the central chemoreceptor to the hydrogen ion concentration? Hydrogen ion concentration is the primary stimulus for the central chemoreceptor. As hydrogen ion in the cerebrospinal fluids and the surrounding interstitial fluid is the only direct stimulus for central chemoreceptors. Unfortunately, hydrogen ion in the blood cannot cross the blood-brain barrier or the blood-CSF barrier. Therefore, changes in the hydrogen ion concentration in the blood will not have a stimulating effect on the central chemoreceptor. Instead, the carbon dioxide in the blood stimulate these neurons secondary by changing the hydrogen ion concentration in the CSF. Now, what is the response of central chemoreceptor to the carbon dioxide? Carbon dioxide has a very potent indirect effect. Carbon dioxide crosses the blood-brain barrier and the blood-CSF barrier because it's a small, soluble, and uncharged molecule. So whenever there is increase in the partial pressure of carbon dioxide in the blood, it's accompanied by increase in the partial pressure of carbon dioxide in the CSF and the interstitial fluid in the medulla. Now, carbon dioxide affects respiration primarily by acidifying the CSF and the interstitial fluid surrounding the central chemoreceptor, as carbon dioxide reacts with water of the tissue to form carbonic acid bicarbonic anhydrase enzyme. The carbonic acid dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions have a very potent direct stimulatory effect on the central chemoreceptor. Thus, the carbon dioxide stimulates the central chemoreceptor indirectly. Carbon dioxide serves as a source of hydrogen ion to stimulate the central chemoreceptor, so the hydrogen ion is the actual stimulus. Remember that hydrogen ion in the blood cannot cross the blood-brain barrier or the blood-CSF barrier. So the changes in the hydrogen ion concentration in the blood will not have a stimulatory effect on the central chemoreceptors. It's important to know that the central chemoreceptor do not respond to mild or moderate decrease in the partial pressure of oxygen. And it's important to know that severe hypoxia, if present, depresses the CNS, including the respiratory centers. Remember that CSF has only small amount of protein, 20 mg per 100 milliliter, compared to the plasma that has 7,000 mg per 100 milliliter. So the CSF is not highly puffered as the blood. Thus, a small change in the arterial partial pressure of carbon dioxide results in large changes in the CSF pH, which will have a strong stimulatory effect on ventilation through its effect on the central chemoreceptor. So it's also important to know that 3% increase in the arterial partial pressure of carbon dioxide will double the ventilation. Now coming to the peripheral chemoreceptors. The peripheral chemoreceptors are special nervous chemical receptors present outside the CNS. They include the carotid bodies and the aortic bodies. The peripheral chemoreceptors are contained within small nodules on the aorta and the carotid artery. They include the aortic bodies scattered over the aortic arch and the carotid bodies located bilaterally at the bifurcation of each common carotid artery. They are located quite close, but they are different from the atrial baroreceptor that we discussed earlier in the cardiovascular system. The peripheral chemoreceptors control breathing indirectly via sensory nerve fibers to the dorsal respiratory group in the medulla. Impulses from the carotid bodies are carried along branches of glossopharyngeal nerve, while impulses in the aortic bodies are carried along the vagus nerve. These nerves together known as buffer nerves. It's important to know that the peripheral chemoreceptors have a very high blood flow, 20 milliliter per minute per gram tissue, which is 20 times the weight of the bodies themselves. They are considered as the organs with the highest blood flow per unit mass. Because of their enormous blood flow, the oxygen needs of the cells can be met largely by dissolved oxygen only, and thus they are sensitive to the oxygen dissolved in the physical solution and not to the oxygen bound to the hemoglobin. So the peripheral chemoreceptor therefore monitor the oxygen tension, that's to say the partial pressure of oxygen rather than the oxygen content of the arterial blood. It's important to remember that because of the peripheral chemoreceptors location and their rich blood supply, they are efficient in sensing the changes in the arterial blood gases in the oxygen, mainly the hydrogen ion concentration, and to a lesser extent the carbon dioxide. The peripheral chemoreceptor are the only sites in the body that detect changes in the arterial oxygen and hydrogen ion concentration, but for the carbon dioxide, they are stimulated to a lesser extent. Now, what is the response of the peripheral chemoreceptor to oxygen tension? Hypoxia is the most potent stimulus for the peripheral chemoreceptor. As the arterial partial pressure of oxygen falls below 100 mm mercury, the rate of discharge from the peripheral chemoreceptors starts to increase, but does not affect ventilation until it reaches 60 mm mercury. This range between 100 to 60 mm mercury at which the arterial hemoglobin saturation with oxygen is stable, representing the plateau part of the oxygen hemoglobin dissociation curve. As the arterial partial pressure of oxygen reached 60 mm mercury, the peripheral chemoreceptor rapidly increased their firing rate and ventilation is doubled. Peripheral chemoreceptors are most sensitive to changes in the arterial partial pressure of oxygen in range between 60 to 30 mm mercury. This is the range at which the arterial hemoglobin saturation with oxygen rapidly decreases, representing the steep part of the oxygen hemoglobin dissociation curve. Ventilation increases steadily until it is increased 6 times when the partial pressure of oxygen reached 20 mm mercury. Drop of the partial pressure of oxygen to 20 mm mercury or less has a direct inhibitory effect on the respiratory center. Now, what is the response of the peripheral chemoreceptor to the hydrogen ion concentration? Well, changes in the arterial hydrogen ion concentration due to causes other than the increase in the partial pressure of carbon dioxide, that's to say the causes of the metabolic acidosis, affect the peripheral chemoreceptor. For example, in metabolic acidosis, the increase in the hydrogen ion concentration caused by the addition of a fixed acid to the blood, such as lactic acid during muscle exercise, stimulates ventilation through the peripheral chemoreceptors. The increased ventilation lowers the partial pressure of carbon dioxide and therefore the hydrogen ion concentration content of the arterial blood, which will return the pH of the blood back to the normal. Now, what is the response of the peripheral chemoreceptor to the carbon dioxide tension? The peripheral chemoreceptor responds to changes in the arterial partial pressure of carbon dioxide, but their sensitivity to carbon dioxide is far less than that of the central chemoreceptors. Denervation of the carotid body reduces the ventilatory effect of the hypercapnia by 20%. So, for example, when a normal subject is given a carbon dioxide mixture to breathe, less than 20% of the ventilatory response can be attributed to the peripheral chemoreceptor. Remember that the peripheral chemoreceptor are particularly important in cases of hypoxia where the partial pressure of oxygen is less than 60 mm mercury, in cases of metabolic acidosis and alkalosis, and in cases of depression of the central chemoreceptors. It's important to know that approximately 85% of the resting chemical drive of respiration results from the stimulatory effect of carbon dioxide on the central chemoreceptors. The remaining 15% come from the stimulatory effect of oxygen on the peripheral chemoreceptors. This will bring us to the important question: why is oxygen lack a weaker stimulus for respiration than the carbon dioxide excess? And the answer is hypoxia has stimulatory effect on respiratory center through the peripheral chemoreceptors, while depress the respiratory center by direct action. So the inhibitory effect balances the stimulatory effect until the oxygen lack is very marked. That's to say the partial pressure of oxygen is less than 60 mm mercury. On the other hand, carbon dioxide has both a direct stimulatory effect on the central chemoreceptor and a reflex stimulatory effect on the peripheral chemoreceptor. Also, the drop in the partial pressure of oxygen from 100 to 60 mm mercury doesn't affect the percentage saturation of hemoglobin with oxygen or the oxygen supply to the tissue. Thus, normally, the oxygen regulation of respiration is not needed, while the partial pressure of carbon dioxide must be regulated almost exactly at 40 mm mercury, as the least change in the carbon dioxide will affect the chemical reactions of the cells. Adding to that, the stimulatory effect of oxygen lack is opposed by the carbon dioxide and hydrogen ion feedback control mechanism, as once ventilation is stimulated by oxygen lack, the carbon dioxide is washed out, and the decrease in the partial pressure of carbon dioxide will depress the respiratory center and counterbalance the stimulatory effect of lack of oxygen. Also, due to hypoxia, the arterial blood will contain greater percentage of hemoglobin in deoxygenated form, which is a weaker acid than the oxygenated hemoglobin. Thus, hypoxia results in some decrease in hydrogen ion concentration of the arterial blood, and this decrease in the hydrogen ion concentration tends to inhibit the respiratory center. Always remember that the increase in the hydrogen ion concentration is the main stimulus for the central chemoreceptor, yet they are stimulated indirectly by the increase in the partial pressure of carbon dioxide. On the other hand, the decrease in the partial pressure of the oxygen, which is a state of hypoxia, is the main stimulus for the peripheral chemoreceptors. To summarize the chemical regulation of respiration, we say that if there is a stimulus that disturbs the homeostasis, leading to increase in the partial pressure of carbon dioxide, increase in the hydrogen ion concentration, or decrease in the partial pressure of oxygen, these are the stimuli that can be detected by the central and peripheral chemoreceptor. These receptors will send the information to the respiratory center in the medulla. The respiratory center will send the order to the effector organ, the diaphragm, and the respiratory muscle, leading to hyperventilation in an attempt to decrease the partial pressure of carbon dioxide, the hydrogen ion concentration, and to increase the partial pressure of oxygen, thus returning homeostasis back to the normal. What is the clinical application of today's session? From this session, we know that the peripheral chemoreceptor monitor the oxygen tension, that's to say the partial pressure of oxygen, rather than the oxygen content of the arterial blood. Consequently, the peripheral chemoreceptor are not sensitive to changes in the oxygen content of the blood caused by anemia, methemoglobin, carbon monoxide poisoning. Thus, in this situation, ventilation is not stimulated because the oxygen tension, which is determined by the amount of dissolved oxygen, remains normal. However, the peripheral chemoreceptors are stimulated when the blood flow decreases markedly, as in cases of severe hemorrhage and hypotension. From this session, we know that approximately 85% of the resting chemical drive of respiration results from the stimulatory effect of carbon dioxide on the central chemoreceptors, and the remaining 15% come from the stimulatory effect of oxygen on the peripheral chemoreceptor. But there are certain conditions under which hypoxia is the major controller of breathing. These conditions include the depression of the brain or the respiratory center by the narcotics or the anesthesia. The effect of carbon dioxide on the central chemoreceptor is abolished. Thus, the effect of hypoxia on the peripheral chemoreceptor becomes the driving force for respiration. If such conditions are treated by giving pure oxygen, both the alveolar and the arterial partial pressure of oxygen will increase above 100 mm mercury. Thus, the driving effect of oxygen lack on respiration is abolished. That's why during oxygen therapy, patients must be watched closely to ensure that the improvement of the degree of hypoxia does not depress ventilation. This problem can be avoided by putting the patient on artificial respiration. By this, we finish today's session. I hope you enjoyed it. Thank you.